So hello, everybody. Welcome to our live stream today. My name is Stacy Leonard, and I'm the events manager here at Connecticut River Conservancy. And I'm really pleased to welcome you to the first episode of our fourth season of our live stream series. In today's episode, Plastic Pandemonium, Source to Sea to Solutions, we'll be talking about the impacts of plastics in our water bodies and feature the ways that CRC is addressing the problem of plastics, from river cleanups to microplastics monitoring to region-wide advocacy. We really appreciate you coming out to learn about this problem and sharing your concerns and solutions together. Before we go on, I'm going to share the usual housekeeping details. This episode is being recorded and it will be available for view within the next 24 hours on our live stream page as well as our YouTube channel. And I'll be sending an email out with the direct links to that, as well as a list of resources and other topics pertinent to this discussion. There will also be a feedback survey, and we invite you to share your thoughts on the presentation as well as ideas for future episodes. Most importantly, we encourage your questions, and we will be saving time at the end to get to them. So please type them in the chat box, and we'll get to as many as possible. And some of us are available to stay on for maybe 5 or 10 minutes extra if the questions are plentiful. So just a reminder to please stay muted while we have the presentation. So the topic of plastics is very meaningful to me for several reasons. I have been the coordinator of our annual Source to Sea cleanup for the past 6 years, and have seen the tangible evidence of plastics being collected and thankfully diverted from our waterways, as I'm sure many of our listeners today have as well. I also live in the town of Leverett, Massachusetts, where we have a very active energy committee that has been bringing the plastics conversation to our community's attention, compiling a list of resources of plastic alternatives, and most recently screening the film, The Story of Plastics, which made me screaming mad and ready for action. And I will share a link to this animated short with you later. Many of us know that our world is overflowing in polluting single-use plastics, primarily made from fossil fuels, and including an enormous amount of chemical additives, some of which are known to be harmful. We may also be familiar with images of plastic waste in the bellies of marine life, and the fact that we each consume microplastics in our food and drink and encounter them in the air that we breathe. You may not know that the global plastic production is projected to accelerate in the future as fossil fuel companies create new products due to the decreasing demand for their fuels for electricity and transportation. In the US alone, it is estimated that we generate over 42 million metric tons of plastic waste. This is a statistic from 2018, with estimates of recycling being only 5 to 9%. Instead, most plastic waste in the US is buried in landfills and some is incinerated, most often in marginalized communities, creating more pollution and harm. Or it's shipped to other countries, or it's released into our waterways on a breeze or down a storm drain. Back at home in our watershed, our cleanup volunteers have picked up and documented the collection of tens of thousands of plastic bottles, including mini alcohol bottles and many other data points. This is only half the story, since not everybody tallies their trash, but we will hear more about that later. Today's presenters will dig into this plastic pandemonium, sharing their research findings and where we can all go from here to be part of the solution. So let me introduce our speakers. We'll first be hearing from Dr. Lavender Law, Research Professor of Oceanography at the Sea Education Association in Woods Hole, Massachusetts. Dr. Law will share her organization's decades-long research on microplastics in the ocean and her work upstream. Trained as a physical oceanographer, Dr. Law has more than 12 months of sea time on oceanographic and sailing research vessels including in eastern North Pacific and western North Atlantic oceans, where plastic debris accumulates in regions dubbed garbage patches. Dr. Law holds a PhD in physical oceanography from Scripps Institution of Oceanography and a BS in mathematics from Duke University. Our second speaker today will be Ryan O'Donnell, CRC's Water Quality Monitoring Manager, who will talk about our pilot volunteer microplastics monitoring program in the Connecticut River watershed. Ryan joined CRC in 2013 as a lab tech in our Greenfield Water Quality Lab, and since 2015 has coordinated volunteer water quality monitoring programs. He is especially focused on using volunteer monitoring programs to bridge knowledge gaps and build coalitions where watersheds cross political boundaries. Ryan holds an MS in Sustainability Sciences and a BS in Geology from UMass Amherst, and a BA in Classical Guitar Performance from Virginia Commonwealth University. Last but not least, we'll hear from Ria Drozdenko, CRC's River Steward in Connecticut. She will talk about our advocacy work and how you can get involved. Ria holds a BA in Sociology with a Civic Engagement Certificate, as well as an MALS in Social Sciences from Wesleyan University. She has lived in Connecticut for most of her life, is a passionate advocate for clean rivers, and is actively engaging state legislators to pass bills that address plastic pollution, tire dumping, and more. We are so lucky to have these experts joining us today. I will say also that before I turn it over to Dr. Law, there are a couple of other CRC staff on the line today. Kathy Erfer is our river steward in Vermont, and she will be helping with the Q&A, as well as Kelsey Wentling from Massachusetts. So I'm going to turn it now over to Kara. Thank you.
Great. Hello, everybody. Thank you so much for joining us here today. First, I'd like to thank Stacy and the entire Connecticut River Conservancy team for the invitation to participate in this important webinar. It's a pleasure to be here. So I'm not going to spend too much time on introductions, but I will try to give you the very briefest overview of ocean plastic pollution based on my work in this field, which I've been doing for now more than 15 years. So I started out studying ocean physics and ended up studying ocean trash. Next slide, please. So first, I'd just like to give you a brief introduction to my organization. For those who are not familiar, I work for Sea Education Association, or SEA, which is an educational nonprofit organization located not only in Woods Hole, Massachusetts. You can see the little star pointing to our land-based campus on Cape Cod, but we also have 2 oceanographic sailing research vessels that are also our classrooms: the Corwith Kramer that operates in the North Atlantic and the Caribbean Sea, and the Robert C. Siemens, which operates in the Pacific Ocean. So just very briefly, we have been for more than 50 years now teaching high school students and mainly undergraduate students in our sea semester program. They come to us on our campus in Woods Hole for several weeks to prepare for a 4 to 6 week voyage, open ocean voyage on one of our 2 tall sailing ships. And in our programs, they're studying the oceans from a variety of perspectives, of course, scientific, but also from a sort of social science, marine policy, or maritime history perspective. And they also prepare to sail as fully fledged crew members on board the ships. They're the ones who are standing watches and working on all aspects of ship operations. The mission of the ship is not only to get from point A to point B, but it's also to collect data to support undergraduate research that the students design on our campus and then carry out while at sea. So we've been sailing the oceans for 50 years. And when I first was pretty early on in my time at SEA in about the mid-2000s, we started hearing these mentions of the garbage patch, the North Pacific garbage patch in the eastern North Pacific Ocean. And in fact, I was about to sail a trip from Hawaii to San Francisco, and a request got patched to me from a journalist. A journalist had called SEA, learned about where we were operating, and asked if we would be sailing through this garbage patch. I didn't have any idea what this person was talking about and tried to do a little bit of research, but Not long after, next slide, please. I think we all probably heard a lot about what is now a pretty common term, the garbage patch. The garbage patch turned into kind of a phenomenon of its own. It was described as twice the size of Texas and 3 times the size of France, as having more plastic than plankton, and at its peak was being described as this floating island visible from space, conjuring up images of plastic bottles and flip-flops and recognizable trash that we all generate in this giant island that astronauts could look at. So this is a really striking and a very memorable image, of course. But let me just tell you first that there is no floating island visible from space. Next slide, please. Here's my very brief data from space. This is a photograph actually taken from the International Space Station by my good friend and former graduate student colleague, Megan MacArthur, who went on to become a NASA astronaut. And she took this image. You have to trust me because there's no land boundaries in this image, but this is in fact the Eastern North Pacific where the so-called garbage patch exists. Next slide, please. So my own experience is, of course, back on the planet and often from a ship in regions called garbage patches. So this is more what it looks like to somebody on the deck of a ship. And this is the sort of view that thousands of our students and our staff scientists and crew at SEA have been observing for decades. So when you're sailing in the subtropical latitudes of the North Atlantic or the North Pacific Ocean, you typically see not much more than water and clouds, and maybe if you're lucky, some albatross or dolphins or flying fish. Next slide, please. But you will, in these parts of the ocean, which is quite different from other areas that SEA has operated, also see floating debris items drifting by, and they could be as frequent as 1 item per minute. So sometimes you recognize them. That's a boot floating upside down in the upper left and a 5-gallon bucket in the lower right photo. More often than not, though, these are sort of chunks of items or sort of unidentifiable pieces. So you can see there's this sort of large mass of plastic film next to the bucket. And in the upper right photo, you see lots of different chunks and bits and pieces of things, in addition to actually recognizable food packaging with Japanese writing on it. And that's because that collection of debris was collected by our students and staff and crew in the Eastern North Pacific about 1.5 years after the very tragic Japanese earthquake and tsunami. So that's also where that tire on the bottom left came from, and very, very clearly from a specific event. Although most of the time we can't actually tell where most of this sort of junk came from. Well, if you're an open ocean sailor or a seagoing oceanographer who spent a lot of time offshore, seeing these items hundreds or thousands of kilometers from land is first extremely surprising. You know, what is this tea kettle or tire or children's toy doing all the way out here? And it's shocking, and then it's sort of depressing because nobody wants to see trash floating around in the middle of the ocean. But the picture changes. Next slide, please. If you measure what's floating at the sea surface in these regions by towing a plankton net through the water for just 30 minutes and then sift through its contents. And this is what our students have done, thousands of them, twice a day, every day. Again, originally, well, not originally, still to collect data to support their research projects. So what you're seeing here are some images of what we call the Newston net. It tows alongside the ship, as you can see in this bird's eye view on the left. It's easily handled by a single student who just deploys it over the side, and then we tow it half in, half out of the water. So it's really sampling the very sea surface. And then when it's come back aboard after 30 minutes of towing, or about 1 nautical mile of sampling, we pour the contents of the net into a sieve, and then students spend time picking out not only organisms of interest for their particular research projects, but also something that looks like this. Next slide. We find, especially in these regions in the subtropics, lots of tiny bits of plastic that we now as a society refer to as microplastics. This is, let me tell you, a particularly contaminated net tow. They don't all look like this, but I show it because it really gives you a sense of the kinds of particles that we're finding floating at the sea surface. So you can see that most of the particles are pretty easy to tell apart from natural organic matter. And that's because they're brightly colored, they're irregularly shaped, and they're kind of have this hard texture if you pinch them with tweezers like you see here. Most of what you see are pieces that are broken apart from items that were once larger items. We know that sunlight weakens plastics. It makes them chemically degrade. And then when they experience shear and stress forces, sort of bending and pulling and grinding in the environment, those items break apart into smaller and smaller pieces. So at SEA, what we'd known for years, in fact, for decades, before the garbage patch hit the scene and hit the internet, we knew that microplastic pollution was pervasive in parts of the ocean that we had sailed and sampled. Next slide, please. So what I'm showing here is a global map of all of the floating microplastics data that SEA has collected since the 1970s. We do have the largest and the longest data set of its kind, and this is really due to the efforts of decades of undergraduates and the folks that were teaching them. So what you can see is each dot is the location of a single plankton net toe, like I showed you in those photos a couple slides ago. And the dot is colored according to the amount of plastic that was measured in that single net toe. So the darkest blue is zero plastic, and then the warmest colors are the highest values. And the map is layered to sort of highlight the highest values. But what you can see is that there are two regions in the Northern Hemisphere centered at about 30 degrees north, one in the Eastern North Atlantic and one, Eastern North Pacific, and one in the Western North Atlantic, that have the vast majority of the high concentrations that we've measured. And this is in fact what the garbage patches are. There are these areas of the ocean where the ocean currents actually sort of come together and slow down based on well-known ocean physics, and causing material that's floating at the surface, like these microplastics, to accumulate. So there's a good understanding of why we see the highest concentrations of these small bits of plastics in these areas of the ocean. Now, I should say there are also similar, quote, garbage patches of floating microplastics in the Southern Hemisphere, also at 30 degrees south. Our ships have just never sampled there. So there are some other expeditions that will show you a similar high concentration in those Southern Hemisphere subtropical ocean gyres. Okay, so next slide, please. So microplastics are not simply floating on the surface of the ocean. We've looked lots of different, many scientists have looked at many different parts of the marine environment. And what we found is that microplastics are the most abundant type of plastic debris and the most widespread. So microplastics have been found mixed with beach sands. That's a photo from Hawaii. Floating at the sea surface. That's another photo from SEA. But they've also been found buried in deep sea sediments and even frozen into Arctic sea ice. But microplastics aren't only in the marine environment. They've also been now found in freshwater environments, as we'll talk about more here, including lakes and streams and rivers. They've been found in the terrestrial environment, including agricultural and other soils. They've been found in the air, in snow, in drinking water, in bottled water, in wastewater, in stormwater. By and large, it seems that the smaller the particle, the more abundant and the more widespread they are. And that's especially true for microfibers. You can see some photos of microfibers in that Arctic sea ice slide there. They're very, very fine, elongated particles. And microfibers are probably the most widespread because not only can they be moved around the environment in water, but they can also be carried easily in the air. Next slide, please. So microplastics are definitely small enough to be ingested by animals from the top to the bottom of the marine food web. And you see some images of zooplankton on the left and barnacles and fish. But we found microplastics really across the entire marine food web. And in fact, plastics ingestion has now been reported in scientific articles for more than 1,200 marine species. So the plastic particles themselves can pose risks to individual animals, maybe causing them to feel full, or if they're small enough particles, there are concerns that those particles can transfer from the stomach or the GI tract into the tissues of the animal, potentially even into the bloodstream. But there's also concern about the chemicals that are associated with these plastics, and that's what that schematic in the bottom right is meant to indicate. So we know that plastics, which is a really broad class of material types itself, is made even more diverse by thousands of different chemical additives. So chemicals that are added to give the plastic its properties specific to its application. But they also can be byproducts of manufacture. And then while drifting around in the ocean, some of these chemicals are actually persistent contaminants that are already in seawater that would then rather stick to the plastic. So the question is, of course, well, what is the evidence of harm if animals are encountering these plastics, and especially if they're eating them or bringing them in through their gills, for example? There is certainly evidence of harm of microplastics, but it's not definitive. It's not easy to make generalizations because the results are typically done in a lab where you have to restrict the analysis to a particular animal and a particular small set of plastics with particular small set of chemicals, and then look at a wide variety of different effects that might be occurring on the animal or on the level of biological organization that you're looking at. So I'd say there's still a lot of work to be done. There is serious concern about this interaction. But right now, I think the field is really focusing on the smallest particles, those smaller than 10 microns in size, which are really difficult to isolate and quantify in environmental samples, but are likely, or at least hypothesized, to pose the greatest risk, again, because they could potentially transfer sort of within the tissues of an animal. Given all that uncertainty, I will say the clear scientific consensus is that we know enough about the risk and harm to marine life and the marine ecosystem to research feasible, effective solutions to stem the continued influx of both microplastics, but also large debris. And there, the evidence is clear when you think about things like entanglement. But I think the focus is, yes, we need to know more about those specific impacts to understand the details, but let's also put some of those resources to understanding the solutions and how to stem that input. Next slide, please. Further, I want to acknowledge that really only very recently are we recognizing that it's not just marine life or even wildlife that are at risk of contamination by plastics, but a wealth of studies are now documenting microplastics in the items that we consume. So starting with seafood, and sea salt, so kind of food products from the ocean. But research has also found microplastics in bottled water and tap water and beer and honey and tea and even fruits and vegetables. We also know that there are essentially microfibers in the air everywhere. And we know that because folks who are doing laboratory studies have a really hard time with contamination from these particles that are just around us all the time. And so these, of course, pose another potential risk if they are inhaled. So we have had some recent medical studies in the past 2 or 3 years that have indicated that, yes, humans are exposed and we're taking in these particles. There have been studies identifying microplastics in human poop and placentas and lung tissue and blood and breast milk. But these studies, much like many of the first studies in marine organisms, are really documenting the presence of these chemical-laden particles, but we can't yet say what the responses within our bodies are to these. But I think it's very clear to say that attention is rapidly shifting to human health questions, and in that sense, the stakes are getting higher. Okay, last slide, please. I just want to show you one more map showing a different distribution of microplastics in the environment globally. And this comes from a citizen scientist initiative led by a group called Adventure Scientists. So in this initiative, volunteers across the globe were asked to collect 1 liter samples of water. So you see the little 1 liter Nalgene bottle on the left, from marine and freshwater systems. And they sent these to Abby Barrows in Maine, who filtered them using that little rig that you see or something like it, and then analyzed those particles, analyzed those samples for particles larger than 0.45 microns. So just to give you a little bit of a sense of what was being studied here compared to our Newstone nets, the smallest particle that could theoretically be measured by this filtering method is about 750 times smaller than the smallest particles collected in our typical plankton net tows by our students, like I showed you earlier. So for that reason, you can see that the distribution of plastics where you see sort of highest values. So here, each orange bubble shows you where a sample was collected that contained plastic, and the bigger the bubble, the more plastic was found. The yellow dots show you where there was no plastic. So the concentrations vary widely from 1 or 2 particles per liter to more than 500, but you can see that there is not the same clear pattern of accumulation like we saw due to the ocean currents for the larger particles. So a couple of things to remember. One, a microplastic is not a microplastic is not a microplastic. This is a really diverse class of contaminants because of material type, because of size, because of chemical composition, and that material, that class of contaminants actually changes in the environment. They become smaller, they chemically change, and they change shape. So it's a hard problem to address when we think about impacts. And And this map also shows you that microplastics are not just an ocean problem. Many of these observations that the citizen scientists took came from freshwater ecosystems, including rivers. And so with that nice transition to rivers, I'm going to hand over the baton to Ryan, who will talk more about CRC's work.
All right, thank you so much. As Stacy mentioned, I coordinate the water quality monitoring programs here at the Connecticut River Conservancy, and we have just are wrapping up our second year of a pilot microplastics program. This is the first program that's looking at microplastics within the Connecticut River watershed. And I would be remiss if I didn't say that my colleagues Gabrielle Chevalier and Aliki Fournier have really made this program possible by coordinating the volunteers and taking the lead in developing the standard operating procedures in the lab by reviewing other programs in other regions doing similar work. So we did a preliminary analysis of the data that we've collected so far to present at the Northeast Biologist Conference about a month ago, which is what I'll be discussing here. And so we have already started having lots of great conversations about our work and looking forward to improving the program as we continue to build it. So a little bit about how the program worked. We collected at 13 locations throughout the watershed, most of them in Massachusetts. And these samples were collected from flowing water, so we weren't sampling the sediment. We were just seeing what are the microplastics in the water column itself. We focused in Massachusetts because most of our funding for the first year came from a grant from the Massachusetts Environmental Trust, but as we've seen the success in this program, we hope to expand throughout the watershed. So at each site, which there's a map on the right side of the slide, volunteers collected samples in a 1 liter glass jar. We used glass to try and minimize the amount of microplastics we were introducing in the process. And we asked volunteers to collect twice each season, so a total of 4 sample events. Some sites only were sampled once the first year, and I have to confess that I adjusted the fridge in the lab too cold and froze and broke several of our samples from this year. But we have at least 3 samples from each site so far, and we'll be collecting more in the future. So you can see our setup for filtering the samples in the middle image of the pictures there. And so we filtered them with a vacuum pump and then removed the filter and examined the filter under a microscope to identify the microplastics. We broke them down by size and category. And so I'll discuss that in the coming slides. The first year, we just had the slides in Petri dishes, which were covered, but they needed to be kept wet in order to be examined and keep the plastics from flying away when they dried out. And we found that this really wouldn't translate well to having volunteers do lab work, which is one of our goals to get that started eventually, because we have a lot of great volunteers and some of them would like to be in the lab more. So for the second year, we were able to successfully adapt a technique from some other groups, which used forensic tape, which is the kind of tape they use to lift like fingerprint dust at crime scenes. So we used forensic tape to fix the filter to a glass slide, which you can see in the bottom right. And this made it so that we could view the slide, essentially an infinite number of times. All the microplastics would be in the same configuration. And so we could do the quality controls necessary in order to start incorporating volunteers in the lab. And then there's a couple pictures from under the microscope, but they're pretty tiny. But yeah, so and then we also did do quality control samples, including field and lab blanks. This is when we fill a sample container with water that hopefully doesn't have any microplastics in it, although that's probably pretty hard to find. But and then we analyzed those as well, and we did find that we were introducing a very small amount of contamination to the samples. But another advantage of starting to use that tape in the processing is the second the tape's applied, we're no longer introducing any new microplastics to the slime. So I just want to show you, I'll do a brief overview of what the first 2 years of data look like, and then all this information is available on our website, and Stacy will send out that link after the live stream. Oops, next slide, please. So this is a look at all of the microplastic data we collected, and perhaps unsurprisingly, it's completely dominated by fibers, and mostly blue fibers. And if you think about what comes out of your lint trap in your dryer, it tends to be blue fibers. So we're thinking that that's probably where a lot of these fibers are coming from clothing. There's a lot of different other colors. I'll talk a little bit about them more, and this shows the different size distribution. We also, in addition to fibers, observed pieces of filament or rope, film, so something kind of like flat and mostly clear, a fragment, so like a chunk of plastic, pellets or microbeads, and then sponge or foam. And those are the categories we looked for. And we found all of them. But like I said, mostly fibers, and a lot of them are blue. Next slide. Since that chart was dominated by the blue fibers, I wanted to look at what's the distribution of the different colors that we find in the different types. So starting with the fibers, over like 60% are blue, and then black, which could possibly be dark blue. But we also saw a bunch of other colors: purples, grays, pinks, red. In the filament category, I think it's notable that we saw mostly transparent filaments, and the notes on the lab analysis suggested that these were probably fishing line. That's what it looked like to the people analyzing the samples. Films had a good distribution of colors. Fragments, once again, dominated by blue. Pellets and microbeads were, either blue to black, white, or clear, which is what most microbead colors are. But then also notable, we saw a lot of white sponge or foam, and the notes from the analysis suggested that it was probably styrofoam. So that's also not surprising, as there's lots of things in and around the water made of styrofoam, and it breaks down into those little pieces. Next slide, please. So this is broken down. I'll answer Kathy's question. So a microbead is a little like round bead of plastic, but some like beauty products and other things have them in them as an exfoliant. So if you get like a face wash with a scrub, sometimes the scrubbing action in that product is plastic, and then that would go down the drain and eventually end up in the water. But also sometimes the beads or the pellets that we're finding might be part of the plastic manufacturing process, where they bring in a bunch of beads and then melt them down and extrude them into whatever they're going to become. So my last two slides are going to look at the different sites. These are in alphabetical order. And of note, most of these are on the main stem of the Connecticut River. H.O. Cook, the second row furthest to the left, is actually what we designated as our headwater site. It's in a state forest, away from any current houses, although I've sampled that, and there are some some cellar holes in the woods out there. And then also we had the West River Marina as the bottom one. And you'll see that that we found microplastics at every site in every sample. And once again, mostly fibers, which are the dark blue color. Something that I noted while looking at this data is that when there was something else besides the fibers, there tended to be kind of like a clump of it. So like if you look at the Haddam ramp in the top right, we have a big clump of orange, which is the filament or rope. And because the fibers are so pervasive, it's hard to tell if they're clumping together from sources, but it seems like if there's one piece of filament, there's going to be a bunch more pieces of filament in the sample. But as you can see, kind of how many microplastics were in each sample was pretty variable. It was just guaranteed that there would be microplastics in each sample. Next slide. And then this is looking at the different colors, which shows a similar distribution, once again, dominated by blue. But then it seemed like if there were other colors, we were going to see kind of a bunch of things of other colors than blue. And you'll notice, once again, in the Haddam ramp down in Connecticut, this on the top right corner, we have that one with like a bunch of transparent. And that was the one that had that chunk of filament. And so it seems like perhaps there was a chunk of fishing line that was in the water and degrading and releasing these bits of filament into the water column. Also of note, the Podchog ramp, which is located in Northfield, we have that one site. It's in the second row in the middle. We have the one site that has a big chunk of white, and that was a big chunk of styrofoam in the sample, or not a big chunk, a bunch of tiny chunks of styrofoam in the sample. So while there does seem to be these microfibers absolutely everywhere, when we find other bits of things, it seems like they're definitely coming from something nearby because there's more than just one-off sample of a different type. Like I said, this is just our preliminary analysis. I'd definitely like to put this data into geographic context, and we're planning on expanding into the watershed. We've received a lot of really positive feedback. We knew that there were microplastics in our watershed, and but we thought it was important to document that because it's so easy to be out of sight, out of mind with things like this. And we can say with 100% certainty now, there are microplastics in our watershed, and there are microplastics even in our headwater sites away from houses. So we'll keep doing this work, keep posting about it on our website. And I'd like to hand it over to Ria now.
Awesome. Thank you, Ryan. And thank you all for being here today. Today, we've talked a lot about the problems of plastics from big to small. So what's next? What can we do about this problem? Earlier, Stacy talked about the source to sea cleanup as a great way to get involved and help remove plastic pollution from our rivers. But we also want to stop plastic pollution before it starts by advocating for strong policy to reduce plastic waste and prevent pollution. During our cleanup, we have our group leaders do a trash tally like these kids are holding. They count up how many items of each type of trash they find, like how many plastic bags they find, how many takeout containers, how many plastic bottles, etc. So now we have this massive database on all the trash we've collected over the past 23 years. This data is incredibly helpful for our advocacy work. For example, I can say that we found 190,383 pieces of styrofoam docks in our watershed in 2022, or that we found 1,057 mini alcohol bottles just in Massachusetts alone. And so this data helps our legislators and the general public get a better sense of the issue, because when you see one plastic bottle on the side of the road, it may not seem that big a deal. But when you think about how we removed 4,301 plastic bottles from our rivers just last year, that makes the problem a lot more clear. So an example of trash advocacy in action in my state of Connecticut. When you purchase a soda or beer bottle in Connecticut, you pay a 5-cent fee, which you get back once you return the bottle for recycling. Evidence shows that bottle bills are extremely effective at increasing recycling. But our bottle bill was made in the 1970s, so it was time for some updating. At CRC, we pushed for a stronger bottle bill that increased the fee from 5 cents to 10 cents and included other types of bottles like hard seltzer, juices, and even kombucha. So we submitted testimony and encouraged CRC members to do the same. And after a few years of pushing for it, in 2021, a stronger bill in Connecticut finally passed, with the updates now starting to take place. And Vermont has seen a similar success story with a law passed in 2019 banning single-use plastics, including plastic bags and straws. And so on our next slide, in 2023, there are quite a few raised bills in our watershed states that attempt to reduce our reliance on plastic and prevent pollution. So here are just a few examples. In New Hampshire, there's a bill to create a committee to study extended producer responsibility, or EPR. EPR is a policy that holds manufacturers responsible for their products after consumers are done with it, whether that's creating a way to safely dispose of it or paying for its recycling. In Vermont, a stronger bottle bill is on its way. It passed the House and is on the Senate floor now. And there's also a bill that would prevent all of those styrofoam dock float chunks from ending up in our rivers. In Massachusetts, there's a bill to have minimum recycled content requirements for plastic beverage containers, and there's a bill to ban plastic bags. And here in Connecticut, there was a bill for packaging EPR, as well as a bill banning styrofoam takeout food containers. So how can you get involved? On our next slide, we have some action steps you can take. First, reduce how much plastic you use. Move away from single-use plastics whenever possible, reuse whatever you can, and remember that we can't recycle our way out of this problem. You can get involved in our Source to Sea cleanup this September, and please, please, please do the trash tally. I know it can be really cumbersome and take some time, but that data truly makes our advocacy work stronger. Next, reach out to your legislators and let them know about this issue and why it's so important to you. If you're not, make sure you're signed up for CRC's news in your state. Myself and the other river stewards send out action alerts about bills that affect the health of our rivers, and we offer a short sample testimony that you can use. And remember that it's super important for legislators to hear from folks like you. And lastly, please share something that you learned today with your community. And now I'm going to pass it back over to Stacy to take us into our Q&A session. Thanks.
Great. I'm going to stop the share actually. Thank you all for all of that research and years of work and trying to identify the problem and figure out where it's coming from and where it is going. I want to remind folks to please put questions in the chat box because there's a lot of people on the line. We're not going to be able to do raising hands quite yet. So Kathy Erfer, I mentioned before, is our river steward in Vermont, and she is here to read off some questions and ask our speakers to share their answers. So take it away, Kathy.
Great, thanks, Stacy. So the first question I actually posed for Kara, is there a specific ecological niche for feeding or reproduction of certain animals that occurs in the same location where you find the higher density of plastic particles in the ocean? It occurred to me that where those currents come together actually creates kind of a micro ecosystem, and I didn't know if that was very important in particular to certain types of animals.
Yeah, that's a great question. Thank you, Kathy. I don't know if anybody can see me. Okay, there we go. So yeah, the answer is yes. So this region, which we can call garbage patch as long as we don't think it's a floating island, is the subtropical gyre ecosystem. And interestingly, that's actually kind of the desert of the ocean. It's an area that has very low nutrients, also tied to ocean circulation. But that doesn't mean it's devoid of marine life. It just means that these are different animals that are living there that are living in very high nutrient zones. So in terms of specific animals, this is actually a question that a colleague of mine, Rebecca Helm at Georgetown University, is investigating. And she's really interested in specifically the animals that live at that air-sea interface in the Neuston layer. So some of these are animals that you may have heard of, like Physalia, which is oh my gosh, I can't believe I know the scientific name and I'm blanking on the common name. Anyway, there's floating animals that might look familiar, some of them that don't, that are feeding in those regions. And then there are also small mesopelagic fish that actually spend time during the day down at depth, several hundred meters deep, and then they come up to the surface at night and feed. And so they could be eating these plastics and then actually transporting them down deeper into the ocean where other animals might be exposed to them. So what the actual impacts are in these communities is a big question. I don't think we know the answer to that yet, but there's also, large animals that like sharks or turtles that are migratory that pass through these regions of the ocean. So there's sort of resident communities, and then there are also other different communities that are certainly encountering these plastics.
Thank you. Ian asks, can plastics also block sea animals' digestive tracts like polystyrene can?
Yeah, definitely. Obviously, the bigger the plastic, the more likely it is to create a blockage. And we've probably all heard the stories of these whales that have had flower pots and car parts in their bellies and agricultural films. I feel like there's one, every few months we hear a story of a whale that washed up, and when you open up its stomach, you see all of this terrible debris. Of course, it depends on the size of the particles and the size of the animals and how much is there, but certainly that is one of the impacts on an individual animal that's eaten microplastics. And the other sort of different side of the same coin is that it can also make the animals feel full. And of course, there's not any nutritive value coming from the plastics, so they may end up starving.
James asks, are micro rubber compounds found in these samples as well? And he's interested in the contribution by tires that we've learned about more recently.
So I'll start with the open ocean. So we are not finding anything that looks like tire particles, probably for two reasons. One, thousands of kilometers offshore, we're pretty far from tires. But two, I believe those compounds, which are synthetic compounds, are denser than water and sink. So there is actually clear evidence of tire wear particles in freshwater ecosystems, potentially even very close to shore. And maybe I'll just toss that over to Ryan to address whether or not they're finding that in the samples you've all collected.
I don't think that we've seen any. I don't have any notes of anything that looks obviously like a tire, but we're not identifying them to like whether they're rubber or plastic.
Thanks. So a commentary here by Tom Metzner from CT Deep. He points out that several towns in Massachusetts have banned the sale of miniatures or NIPS bottles, but Connecticut state law currently prohibits a municipality from banning their sale. That law needs to be updated. Diana asks, is there an easy way for anyone to send water samples from personal wells or local rivers to a lab for microplastic testing? Is that affordable, and would it help contribute to regional data gathering?
I mean, from CRC's perspective, we don't have the capacity to just accept samples outside of specific programs. They are pretty time intensive, and we're certainly not certified in microplastics analysis. I don't know much about groundwater testing for microplastics. I haven't looked into that yet, or know about regional community science efforts, but that's certainly interesting. Kara, I don't know if you know about anything like that.
Yeah, so there certainly are labs that can analyze samples, but it's very expensive. And I think we're really we're not yet at the point where we're doing environmental monitoring, right? It's still sort of looking to kind of, as Ryan really nicely described, looking to say, yes, they are here in our community. We need to start thinking about these and figuring out, you know, baseline concentrations. If we take certain actions, are they going to decrease over time? Ultimately, that will require sort of environmental monitoring, like you've described, I think, in the question. But right now, it is really time intensive. And in fact, if you know, if you're looking at the size particles that are being collected in these 1 liter samples, typically you actually have to go another step and do a spectroscopic analysis to identify that the actual chemical composition, to make sure that it's a plastic and not a cotton, a wool, a blend, because so many of these are likely textile fibers. And there can be natural particles that also, you know, kind of, you might think look like plastic. So we're not quite there yet. California is ahead of the game. They do have now a program that's really critically looking at drinking water, and they're trying to develop some standard protocols for measurement. I don't think we're there yet at the monitoring stage. They're sort of trying to figure out what is a safe level of microplastics in drinking water. So we may be moving there as we go forward, but at this point, unfortunately, it is really a sort of manual grinding exercise, like Ryan and the team of volunteers have experienced. And thank you to Lee for Portuguese Man of War as the Visalia.
Awesome. Thanks for translating that. I was like, why did he put Portuguese Man of War in there? So, Becky asks, regarding clothes containing plastics, are fibers released mostly during laundering or just with regular wear? And any sense of how effective Cora balls and special bags, etc., are to wash clothes in?
I'm happy to take a stab at that unless there's somebody else that wants to. So yes, we certainly know that textiles release fibers during the washing cycle. There have been scientific publications that document the number of fibers released depending on what kind of, you know, fabric it is, if you have a front loader or a top loader, what kind of detergent you use. There are also studies looking at those sort of intercepting devices like the Cora ball. There's a variety of products now that you can install in either the Cora ball goes into your washing machine with your clothing. There's others that you can put in line that filter the wastewater as it comes out of your washing machine. And they certainly trap fibers that would otherwise go into wastewater. They don't trap all of them. There have been some studies looking at the effectiveness of, you know, X, Y, or Z filter. The dryer, we know, right? We all have lint filters that we empty. So the washing machine filter is not dealing with anything to do with the dryer or the dryer vent that goes out into probably your backyard if you have a dryer at home. And one thing that's really important is that you have to think about how you're cleaning those filters. So this is something easy we can all do, and we can reduce the amount of clothing fibers going into wastewater or the environment. But I think for sort of the long-term solution to textiles, it's going to probably require better engineering of fabrics so that they shed less, so that we can wear them longer, right? I mean, and back to sort of Ria's first point of what you can do is reduce. And so fast fashion is getting a lot of, you know, finger pointing of buy, use, and dispose quite quickly. I think the longer that we can wear the items that we own, and especially if they're built that way to not be losing their fibers, that's sort of the longer term solution.
And Becky also asks about, are there other EPR laws in effect in the US, and how good is the New Hampshire legislation? Which I can actually respond to a little bit. There are various states have different numbers of extended producer responsibility laws. Right now, I think Vermont and California are the leaders in terms of the number of laws on the books that are structured in a way that require extended producer responsibility. I can't speak directly to how good the New Hampshire legislation is. I think there's been several attempts to introduce legislation in New Hampshire to consider extended producer responsibility. And so this study bill will, I think, help the legislators spend some time looking at what's been done in other states and how effective EPR is. So definitely a good step in the right direction. New Hampshire does actually have an existing extended producer responsibility law, I think on mercury, the mercury plugs or things that you find in automobiles. If I am not mistaken, I'd have to look it up. That was all the questions we had. Got a couple minutes if anyone else has some burning questions you want to put in the chat. Tom adds that, California, Maine, Oregon, and Colorado have packaging EPR laws, which I think all passed in the past year or two. So.
I just want to jump in since we're getting close to the end of the hour, but continue to encourage you to add your questions to the chat. And if you want to stay around for a few minutes after 1 o'clock, we can maybe turn people's cameras on and people can raise their hands. But I mostly really want to thank everybody for coming out today. It was a very, very stimulating discussion. And especially thanks to Dr. Kara Law, Ryan O'Donnell, Ria Drozdenko, and Kathy Erfer on the questions, and for all of you for sharing your expertise and your time. And hope that everybody is leaving feeling inspired to sort of roll up their sleeves and get involved. I want to also just mention we have another live stream in the works on June 14th. It'll be on the topic of fish migration and various related topics. So we hope you'll come out for that. And you can find all of our events listed on our events calendar, which is ctriver.org/events. And I can throw that in the chat, and also it will be in the email that you'll get later. So thanks again, everyone, for coming. And I see there are some more questions coming in, so I'm going to shut up and maybe we could take 1 or 2 or 3 more. Thanks, everybody.
Someone asked how you can volunteer other than the river cleanup days, and I did stick a link into our website that has some other opportunities for surveying fish nests, things like that, water quality sampling. Some thank yous. Someone is Valerie is looking for, as an elementary teacher, most of the curriculum is geared toward high school. Does anyone know of somewhere where materials for elementary students around this topic? So any suggestions for that? And Carrie responded to check the NOAA Marine Debris Program.
Yeah, I'm also going to put the link to our SEA Plastics Lab website. We do have a few activities that are suitable for students of different ages if under our resources tab. But if anybody's interested more in the research we do or some of those kinds of resources, please check it out. Thanks.
And I think we hit them all of the comments and things. So.
Okay, well, wonderful. So I can't see if anybody else is out there. Thanks everyone for being here. And you will get an email with the contacts of all of our presenters as well. So you can ask any follow-up questions that way too. So thanks again. We'll see you next time. Bye-bye.